Welcome to another deep dive. We've got quite the collection of scientific excerpts to explore today, ones that touch upon, well, pretty diverse areas of biology, if I'm honest.
Diverse is an understatement, wouldn't you say?
Yeah, maybe. I mean, we're going from like the microscopic world of DNA and protein synthesis
Yeah.
all the way to the complex signaling pathways inside our brains. It's a lot.
It really is like taking a journey through the building blocks of life itself. We're talking Nobel Prize lectures and cutting-edge research papers
Ooh.
focusing on telomeres,
Okay.
ribosomes,
Mm-hmm.
and even how our neurons, actually talk to each other.
Oh, I love this already. Get ready for some aha moments, folks.
Oh, there will be plenty of those, I assure you. The connections between these topics, they might seem surprising at first.
Right.
But trust me, as we go deeper, you'll see just how beautifully woven this tapestry of life really is.
Okay, I'm hooked. Let's dive right in. First stop, the world of telomeres, those protective caps at the ends of our chromosomes. And we've got this incredible quote from Elizabeth Blackburn's Nobel lecture, NBL 48.pdf, to kick things off. She says, "One of the first things that I wanted to do with the new linear plasmid with two tetrahamina ends was to use it as a vector for cloning natural telomeres from the ends of yeast chromosomes." Whew, what a mouthful.
It's a mouthful, but it beautifully captures the essence of scientific curiosity, doesn't it?
Yeah,
She was essentially trying to figure out if these protective caps were, universal.
universal.
Yeah, like could you take the plastic tip from one shoelace and put it on another from a completely different shoe?
Okay, I'm seeing the analogy. So what did she find? Was it a simple swap?
Well, not quite. Her research, it was successful,
Mm-hmm.
but it revealed something pretty unexpected about yeast telomeres.
Hmm.
They had this irregular sequence of DNA bases. G13T repeats, to be precise.
G13 what?
G13T repeats. And that was a head-scratcher because it was different from the, very specific repeating sequence they'd seen in tetrahemida telomeres.
Okay, time for a quick translation. For those of us who don't speak fluent DNA, why was this irregular sequence such a big deal? Like, what's the implication here?
Think of it this way. You're putting together a puzzle, right?
Hmm.
And you find a piece that kind of fits, but it has these weird bumps and curves.
Oh,
It makes you realize that the puzzle is way more complex than you initially thought.
I see.
This irregular sequence in yeast telomeres, it basically challenged the existing understanding of how telomeres are structured, and how they actually function.
So not just some obscure yeast detail. This has got to have some connection to us humans, right?
Absolutely. Every time a cell divides, its telomeres get a little shorter.
Right.
It's like a biological clock ticking away.
I've heard that.
And when those telomeres get critically short, well, the cell can no longer divide properly. That contributes to aging and all those age-related diseases we'd rather avoid.
So these irregular sequences, could they be affecting how fast those cellular clocks are ticking? Like, could they be a factor in how we age?
Now, that's a fantastic question, and it's one that researchers are actively investigating,
Hmm.
Understanding these different types of telomere sequences and how they work could totally change how we approach aging and age-related diseases.
Whoa, you've definitely got me thinking differently about those tiny caps on my chromosomes. Let's shift gears a bit, though.
Okay.
Our next stop is the world of ribosomes, those protein factories of our cells,
The ribosomes. We're diving into NBL 57.pdf for this one,
Okay.
which explores this fascinating thing called antibiotic synergism.
Synergism.
Yeah. Some antibiotics, when you use them together, become way more effective at killing bacteria than when used alone.
Interesting. So, like, they team up.
Exactly. It's like they coordinate an attack.
Okay, I'm intrigued. Break it down for me. How do these antibiotic partnerships work?
All right, picture this. The ribosome is like a bustling factory with multiple assembly lines, loading docks, the whole works.
Okay.
Now, one antibiotic, let's say a streptogrammin, comes in and blocks the main assembly line, preventing the production of essential proteins, like hitting the factory's core operations.
Gotcha. And then what? Another antibiotic swoops in and sabotages the loading dock, stops the delivery of raw materials.
You got it. That's where the second antibiotic comes into play. It blocks the exit tunnel of the ribosome, so the newly made proteins, they can't leave the factory. This double whammy, this dual attack, it cripples the bacterial cell, making it much harder for it to survive.
So it's like a pincer movement,
Oh.
attacking from both sides. But this research also mentions something about the ribosome being incredibly flexible.
Right.
What's that about? Is the factory like rearranging its layout mid-attack?
That's a great way to put it. Ribosomes can actually reshape themselves, especially the peptidyltransferase center.
The what?
The peptidyltransferase center,
Yeah.
the PTC, it's that main assembly line we talked about. It's like the factory sensing the presence of the antibiotics and subtly shifting things around to make it harder for the antibiotics to actually bind.
Wow, that's some serious adaptability. But look, doesn't this flexibility make it trickier to design new antibiotics? If the target's always shifting, how do you hit it?
That's the challenge for sure. And it's super relevant when you look at chloromethylene antibiotics. These target the highly conserved PTC in bacteria, aiming to disrupt protein production.
But isn't the PTC also present in our own cells? Wouldn't attacking it harm us too?
Good point. That's the tricky part. But here's the clever bit. These antibiotics exploit the fact that bacterial ribosomes are a bit more flexible in the PTC region than our own. It's a tiny difference,
Right.
but allows scientists to design antibiotics that selectively target those bacterial factories, leaving our own cells relatively unharmed.
It's amazing how scientists can leverage those tiny structural differences for targeted therapies. It really shows how important it is to understand not just what these microscopic machines do, but how they do it.
Couldn't agree more. And this deep understanding of ribosomal function is crucial as we're facing the growing problem of antibiotic resistance.
Yeah, that's scary.
It's like an arms race, trying to stay one step ahead.
Okay, so from protecting DNA to building proteins, we've covered some serious ground here. But our deep dive doesn't stop there. For our next stop,
Where are we going?
we're zooming in on something called slow synaptic transmission in the brain.
Oh, this is a good one.
Ready to explore how our neurons actually communicate.
Absolutely. Let's dive in. I'm excited to see how we're moving, from the molecular level to the intricate world of the brain.
Yeah, it's quite the leap.
And this next source, NBL49.pdf, gives us this perfect visual. It's a detailed image of, some of the signaling pathways involved in slow synaptic transmission.
I'm picturing like a complex wiring diagram.
Exactly. With arrows and labels all over the place.
Okay, so this isn't just about how fast signals travel, right? What makes this slow communication so important?
Good question. It's less about speed and more about control, really. Modulation. You see, communication between neurons, or synaptic transmission, if you want to get technical, can happen in two main ways, fast and slow. Fast synaptic transmission is like the express lane, responsible for those rapid responses,
Right.
like reflexes or processing sensory information really quickly.
Got it.
But slow synaptic transmission, that's where the real fine-tuning comes in. It's like having the volume controller, the equalizer on a stereo system.
So instead of just sending the signal, it's about shaping and refining it, making sure the message comes across loud and clear, or maybe even a bit muted.
You got it.
Yeah.
And it achieves this control in a couple of fascinating ways. First, it can regulate what we call the phosphorylation state of various proteins, including neurotransmitters.
So free what now?
Phosphorylation. Think of it like a light switch that can turn a light on or off. Adding or removing a phosphate group can activate or deactivate a protein, changing its function, how it works.
So it's like fine-tuning the individual components of the neural network, making sure each one's working at the right level.
Exactly. And the second way it modulates things is by, controlling the amount of neurotransmitter that's released. Imagine you're sending a text message. You can choose to send a short, concise message or a long, detailed one, right?
Yeah, it depends who you're texting.
Right. Well, slow synaptic transmission is like adjusting the length of that message,
Yeah.
controlling how much information is passed along to the next neuron.
Okay, I'm starting to see the bigger picture, but NBL49.pdf points to something pretty important, the potential for understanding neurological disorders, and it specifically mentions dopamine signaling pathways.
Yes. Dopamine. This is where it gets really interesting.
Right.
Because dopamine plays a crucial role in so many brain functions, from motivation and reward to movement and attention. And disruptions in dopamine signaling, well, they're implicated in a whole range of neurological and psychiatric disorders. We're talking Parkinson's disease, schizophrenia, ADHD, even drug addiction.
So if we can better understand how slow synaptic transmission is tweaking and fine-tuning these dopamine signals, could that lead to new treatments for these conditions?
That's the hope. And there's this really encouraging note in the source that points out that many slow-acting neurotransmitters, not just dopamine, they work by similar principles. This suggests that the insights we gain from studying one pathway could have a huge impact, potentially leading to therapies for a wide range of brain disorders.
Wow, that's amazing. It's like it reminds us that even though these processes are happening on a tiny scale, they have this huge impact on our overall health and well-being.
Exactly. That's what makes this deep dive so fascinating.
I agree.
We're starting to see how these seemingly separate areas of biology, from telomeres to ribosomes to synaptic transmission, they're actually all interconnected.
Right.
And understanding those connections, well, that could have a profound impact on how we approach aging, disease, and even, just the very essence of what it means to be human.
I'm feeling a whole new level of respect for my brain and all its intricate workings.
You and me both. But we've covered a lot of ground already.
I know, right?
We'll be back soon to continue our deep dive into this incredible world of science and discovery. Don't go anywhere.
Stay tuned.
Welcome back. I'm really excited to dig into this next source with you. It's a bit of a detour, taking us from, biology into the world of physics and engineering.
Ooh, a detour. I love those. What have you got for us?
This time we're looking at NBL71.PDF. It focuses on a pivotal moment in the development of blue LEDs. It's the story of Isamu Akasaki and his groundbreaking work on creating high-quality indium gallium nitride layers. InGaN for short.
InGaN layers. Okay, I'm going to need a little help here. What are those, and why were they so important for LEDs?
Think of InGaN as like the key ingredient for generating blue light in LEDs. Before Akasaki's work, red and green LEDs, they were already around.
Yeah.
But blue, that was a real challenge. You see, you couldn't just, mix the existing colors to get blue. You needed this specific material that emitted light at the right wavelength.
And that's where InGaN came in. It was the missing piece of the puzzle.
Exactly. Akasaki, he figured out how to grow these InGaN layers with incredible precision using a technique called two-flow metal-organic chemical vapor deposition. Quite a mouthful.
I'll say.
It was a breakthrough, really. It basically unlocked the potential of blue LEDs.
So was it just about getting the right shade of blue? What made this discovery so significant?
Well, think about it this way. With red, green, and now blue, you have the primary colors of light.
Yeah.
Right?
Yeah.
And what happens when you mix those primary colors? You get white light.
Ah, so this wasn't just about a new color. It was about creating white LEDs, the kind we use in, like, everything now.
Exactly. Akasaki's work, it revolutionized lighting technology. We suddenly had access to, energy-efficient, long-lasting white LEDs that could replace those old incandescent bulbs. It's a fantastic example of how fundamental research, in materials science can have this ripple effect, impacting our everyday lives in ways we might not even realize.
It's like a chain reaction of innovation, all sparked by the quest for a single color. I love how these deep dives always reveal these unexpected connections. Speaking of which, where are we headed next?
Okay, so from the microscopic world of InGaN layers, we're now venturing into the vastness of space. Get ready for supernovae.
Supernovae, those massive explosions that mark the death of a star, right? I have to admit, those always capture my imagination.
They're not just visually spectacular, though. They're incredibly valuable for astronomers. They're like, cosmic laboratories that give us these insights into the expansion of the universe, the life cycles of stars, even the fundamental laws of physics.
Wow, okay. And our source, NBL77.PDF, drops us right into the middle of a supernova hunt, led by Brian Schmidt back in 1995. It describes the discovery of a supernova that was, at the time, the most distant one ever observed. So, how do you even begin to search for something like that? It seems like finding, I don't know, a needle in a cosmic haystack.
It's a bit like playing a cosmic game of spot the difference,
Okay.
They used a technique where they compared images of the same patch of sky taken at different times, and any new bright spot that, that popped up in a later image could signal a supernova.
That's actually pretty clever. But sifting through all those images must have been a monumental task.
It definitely required patience, that's for sure, and a keen eye. But their efforts, they paid off. They found this incredibly distant supernova in a galaxy with a redshift of Z0.48.
Redshift. Now remind me, what does that tell us again?
Okay, think of it like the Doppler effect, but for light.
Right.
You know how the sound of an ambulance siren changes as it, as it gets closer and then moves away from you?
Yeah, the pitch gets higher as it approaches and lower as it drives away.
Exactly. Well, light behaves in a similar way. As an object moves away from us, the light waves that it emits get stretched out, shifting towards the red end of the spectrum. So a higher redshift means the object is moving away from us faster and is therefore further away.
So this redshift of Z0.48, that means the supernova was incredibly far away, even for, cosmic scales.
Billions of light years away. to be precise. And that light from the explosion, it had been traveling for billions of years before reaching Earth. And by studying these distant supernovae, astronomers can sort of piece together this timeline of the universe's expansion.
It's mind-boggling to think about those timescales. But hold on, didn't they also discover something surprising about the universe's expansion?
They did. The work of this team, along with other researchers, it eventually led to the discovery that the universe's expansion isn't constant. It's actually accelerating.
Whoa, seriously?
Yeah, it was a huge surprise. And it actually earned them the 2011 Nobel Prize in Physics.
Talk about a game-changing discovery. It's amazing how these distant explosions can tell us so much about, the very nature of the universe. But let's shift gears again. Our next source brings us back to Earth, but this time it's all about polymers.
From the vast scales of the universe to the molecular world of polymers, what a journey we're on. This next source, NBL89.PDF, dives into this fascinating class of materials called conducting polymers.
Conducting polymers. I always thought of polymers as being, more like plastics, insulators. So what makes these polymers different? Why can they conduct electricity?
It all comes down to their unique molecular structure. Think of it like a chain of repeating units, each with alternating single and double bonds. This creates a pathway where electrons they can move freely along the entire polymer chain, carrying an electrical current. It's like having a microscopic highway for electrons to travel on.
So it's the structure of the polymer chain that enables this conductivity, not the material itself, but how it's put together.
Exactly. And this opens up all these possibilities for these materials. The source highlights how they're lightweight, they're flexible, and they're relatively easy to process.
Okay, those sound like some pretty desirable traits. But how are they actually used in the real world?
Oh, you'll find them in all sorts of applications. They're used in organic LEDs, OLEDs, those vibrant displays you see on smartphones and TVs.
Ah.
They're also used in solar cells, transistors, sensors, even some biomedical applications like, artificial muscles and drug delivery systems.
Wow, they really do it all. But the source also mentions a challenge with these polymers. It says they tend to be insoluble and infusible, which honestly doesn't sound very practical.
You're right. Those terms essentially mean they don't dissolve easily in common solvents, and they don't melt easily. It makes it tricky to process them into, useful shapes and forms.
So it's like having this fantastic building material, but you can't mold it into bricks or beams.
That's a great analogy. But thankfully, scientists and engineers have developed clever ways to overcome this. They've come up with techniques like side chain functionalization and copolymerization, which allow them to, modify the polymer structure and make it more processable without sacrificing its conductivity.
So it's like adding handles or hooks to the polymer chain, making it easier to work with.
Exactly. And these innovations, they've truly unleashed the potential of these conducting polymers, leading to a revolution in materials science and technology. It's amazing how sometimes a little tweaking, at the molecular level can have this huge impact on the macroscopic world.
I love how these deep dives always highlight those unexpected connections, between the microscopic and the macroscopic. But hold on, everyone. We've got even more fascinating sources to explore. We'll be back soon to continue our journey through this world of scientific discovery.
Welcome back to our deep dive. We're about to enter a realm where physics, I think, meets philosophy. The fascinating world of thermodynamics. And specifically, we'll be exploring the thermodynamics of computation. Our source, it's old10.pdf, and it really dives into the energy cost of, performing calculations, and this intriguing concept of reversible computing.
Okay, I gotta admit, the term reversible computing kind of sounds like science fiction to me. Isn't there always some energy loss, some heat generated, whenever we do a calculation?
It's definitely true with our current computers. But reversible computing, it explores this theoretical possibility. What if we could design computers that operate with zero energy loss?
Zero energy loss. That sounds like we're venturing into perpetual motion territory.
It's not quite perpetual motion, but it's a fascinating thought experiment. The source actually describes a hypothetical zero entropy loss reversible computer.
Okay, so help me wrap my head around this. How could a computer possibly perform calculations without generating any heat or losing any energy?
The key idea is, reversibility. Imagine a logic unit that, it performs a calculation, And then this is important. It can reverse that calculation,
Yeah.
essentially undoing the steps and returning to its original state.
It's like hitting the rewind button, not just on the data, but on the energy used to process it.
Exactly. The idea is that, by carefully designing these reversible logic units in operations, you could theoretically recover all the energy used in the computation.
But wouldn't that require, like, perfect efficiency? Isn't there always some loss due to, friction or heat dissipation in the real world?
You're absolutely right. In practice, achieving perfect reversibility, it's, well, likely impossible. But that's not really the point here.
So it's less about building a practical machine and more about pushing the boundaries of what we think is possible, kind of like a thought experiment that could, inspire new ways of thinking about energy and computation.
Precisely. And who knows, maybe these theoretical explorations could lead to, more energy-efficient computing devices down the road.
Okay, I'm starting to get it. So we've gone from those microscopic factories in our cells to the vast expanse of the universe, and now to the theoretical limits of computation. Where does this deep dive take us next?
From the theoretical to the practical, our next source, old19.pdf, delves into the world of differential geometry. It's, a branch of mathematics that deals with curves and surfaces.
Differential geometry. That sounds pretty abstract. How does that connect to the real world?
You might be surprised. Differential geometry is actually the mathematical language of Einstein's theory of general relativity, the one that describes gravity as the curvature of space-time.
Whoa, okay, that's pretty significant. But besides, warping space-time, what else can we do with differential geometry?
It has applications in, a ton of fields,
Yeah.
from computer graphics and animation to robotics, even understanding the shapes of biological molecules like proteins.
So it's a mathematical tool that can help us understand and model, like, all sorts of complex shapes and structures.
Exactly. And our source focuses on some of the key concepts in differential geometry, things like curvature and torsion.
Okay, so curvature, that's like how much something bends, right? But what about torsion? What does that describe?
Imagine a helix or a spiral staircase.
Okay.
Torsion describes how much that curve twists, out of the plane. It's a measure of its twistiness.
So curvature is like bending a piece of wire, and torsion is like twisting it.
That's a great way to think about it. And these concepts, they're crucial for understanding the geometry of curves and surfaces, in 3 dimensions.
And how does all this translate into real-world applications?
The source gives a couple of interesting examples. It talks about, how to calculate the curvature radii of curves, like the epicycloid and the catenary.
Okay, you're going to have to refresh my memory. What are those again?
An epicycloid is the path traced by a point on a circle as it rolls around another circle. Think of like the path of a point on a bicycle tire as you're riding down the street.
Ah, I can picture that. And what about the catenary?
A catenary is the curve formed by a hanging chain or cable, that elegant shape you see in suspension bridges.
Right. So by understanding the math behind these curves, engineers can design structures that are both strong and, well, aesthetically pleasing.
Exactly. Differential geometry gives us the tools to analyze and optimize these shapes, for all sorts of different applications. It's a beautiful example of how math can be used to understand and shape the world around us.
Okay, I'm starting to see how this seemingly abstract branch of math has some very real-world implications. What's next on our agenda?
Our deep dive now takes us into the world of game theory and mechanism design. We're going to explore how we can actually design systems that incentivize people to behave in certain ways.
Game theory. I always associate that with, like, strategy games.
Yeah.
Chess or poker. But how does it apply to designing systems?
You'd be surprised how widely applicable game theory is. It's this powerful tool for understanding, strategic interactions, not just in games, but in economics, politics, even designing things like online auctions or matching markets.
Matching markets. Are we talking about, like, those dating apps that use algorithms to match people based on their preferences?
That's a great example. And our source, all13.pdf, it focuses on this key concept in mechanism design called Meyerson's lemma.
Okay, break it down for me. What does Meyerson's lemma tell us?
Imagine you're designing a system where, you want people to be truthful. You don't want them to lie or, misrepresent themselves. Meyerson's lemma essentially says there's only one way to design a system where truthful reporting is always the best strategy, for everyone involved.
So it's like finding the magic formula for honesty, a way to make sure everyone plays by the rules.
That's a good analogy. And this is super important for designing, auctions, voting systems, other mechanisms where truthful reporting is, absolutely crucial.
But let's be realistic. Isn't it impossible to design a system that's completely immune to, manipulation? Won't people always find ways to game the system?
You're right. Achieving perfect honesty is, well, probably impossible. But mechanism design is all about creating systems that are as robust and manipulation resistant as possible. It's about understanding those incentives that drive human behavior,
Yeah.
and using that knowledge to kind of nudge people towards more desirable outcomes.
Sounds like a delicate balancing act, but a fascinating one nonetheless. Where does our deep dive take us from here?
We're heading back to physics for our next stop, exploring this mind-bending concept, black hole thermodynamics.
Black holes, those cosmic objects where gravity is so strong that nothing, not even light, can escape. What could they possibly have to do with thermodynamics?
That's what makes this so intriguing. Our source, although 30.pdf, dives into this idea that black holes actually have a temperature.
Wait, how is that even possible? I thought black holes were supposed to be like incredibly cold and empty since they suck everything in.
That was the classical view. But in the 1970s, Stephen Hawking made this groundbreaking discovery. He found that black holes actually emit radiation due to quantum effects near their event horizon.
The event horizon, that's the point of no return, right? Where gravity gets so strong that nothing can escape. How could something be radiating from there?
That's the mind-boggling part.
Yeah.
Because of the strange world of quantum mechanics, even seemingly empty space is actually this chaotic sea of virtual particles popping in and out of existence. And near a black hole's event horizon, some of those particles can get pulled in while others escape, and they carry energy away.
So it's like the black hole is slowly evaporating, releasing energy in the form of this Hawking radiation.
Precisely. And this radiation, it has a temperature. And strangely enough, it's inversely proportional to the black hole's mass, meaning smaller black holes are actually hotter than larger ones.
That's counterintuitive. You'd think the more massive something is, the hotter it should be.
You would think so.
Yeah.
But black holes, they operate by a whole different set of rules. And this discovery of black hole thermodynamics, it revolutionized our understanding of these objects. And it raised these profound questions about the nature of information, the fate of the universe, even the limits of, our current theories of physics.
It's incredible how something so seemingly destructive and mysterious can also be this source of such profound insights. Where does this journey through knowledge take us next?
Our next source, it takes us to a more personal realm.
Uh-huh.
It's an excerpt from Randy Pausch's last lecture, old21.pdf. It's this really moving reflection on, life, dreams, and making the most of our time.
Randy Pausch, he was that computer science professor, right?
Yeah.
At Carnegie Mellon, the one who was diagnosed with pancreatic cancer. I remember hearing about his lecture. It was incredibly moving.
It was. And facing his own mortality, Pausch, he decided to give this lecture, not about his research, but about, life lessons.
Yeah.
Achieving your dreams,
Yeah.
overcoming obstacles.
So it was less about like equations and algorithms and more about the human experience.
Exactly. The source captures this, his shift in perspective so beautifully. He realized that, while many were focused on the behavior of commodities, he was way more interested in the behavior of people.
That's such a powerful statement. It reminds us that sometimes the most important things in life, they're not about, scientific discoveries or technological advancements, but about human connection, finding meaning, and making the most of the time we have.
And Pausch's lecture, it resonated with millions of people worldwide. It inspired them to chase their dreams and cherish the people around them.
It's a beautiful example of how sharing our personal stories, even the difficult ones, can have such a profound impact on others. Speaking of personal stories, where does our final source lead us?
Our last stop takes us into the world of fashion design. We're showcasing the portfolio of a student at the University of Cincinnati. It's old38.pdf, and it offers this glimpse into their creative process, their skills, and aspirations.
So we're going from the depths of black holes to the runway. That's quite a transition. What does this portfolio reveal about, the world of fashion design?
It shows the student's journey from, sketches and inspiration boards to finished garments. You can see their attention to detail, their craftsmanship, their ability to translate these ideas into wearable art.
And did the student have any real-world experience? Did they get a chance to, apply their skills outside the classroom?
They did. The portfolio highlights several internships, including one with Guerin Inc., It's a major apparel company in New York City.
Wow, so they got a taste of what it's like to work in the fast-paced world of fashion. What kind of work did they do?
The source describes their responsibilities.
Yep.
They assisted designers with, line plans, organized samples, even helped to develop new lines.
That's impressive. It sounds like they got a well-rounded experience from the conceptual side to the practical side of fashion design.
It's a great example of how hands-on experience and internships can be so valuable for anyone who wants to pursue a career in a creative field.
Sure.
Building a portfolio that showcases, your skills and your passion. That's crucial.
And on that note, we've reached the end of our incredible deep dive. We've journeyed from the microscopic world of telomeres and ribosomes to the vast expanse of supernovae and black holes. We've explored the theoretical limits of computation, the elegance of differential geometry, and the power of game theory. We've even touched on the human side of science, reflecting on life, dreams, and the creative spirit.
It's been an amazing journey, really, through these interconnected realms of, knowledge and human experience. And it reminds us that learning isn't just about, accumulating facts. It's about making connections, seeing the world in new ways, and appreciating the beauty and complexity of both the universe and ourselves.
I always love how these deep dives leave me with, honestly, more questions than answers.
Me too.
It's a reminder that there's always more to discover, more to explore, more to understand.
And that's the beauty of it. This deep dive is really just the beginning. We encourage you to, keep that spark of curiosity alive, to keep questioning, keep exploring, and keep diving deep into this vast ocean of knowledge that surrounds us all. Who knows what amazing discoveries await?
Thanks for joining us on this incredible journey. Until next time, keep those minds curious.